For the last recording on the nematode chapter, we will talk about Trichinella spiralis. This one is unique for several reasons. First, it does not have a common name. It's only known as its scientific name by its genus and species. Secondly, there is no egg in this life cycle. There are only adults and larvae. And third, it's not categorized as an intestinal roundworm like the other ones were. This is categorized as a tissue roundworm. So trichinosis is the name of being infected with the larva of Trichinella spiralis, and you'll see that it prefers to set up shop in striated muscle tissue. The adults do make their way to the intestines, but they produce live larvae, meaning no eggs, so the larvae come directly out of the adults. Then the larvae leave the intestines and enter circulation, where they can go to any area of the body they want to. The larvae have a preference for setting up home in our striated muscle tissue. And once they embed themselves in the muscle tissue, they do mature a little bit, they become infected, but then they're encapsulated by the host tissue. They get stuck here. They don't leave the muscle tissue after this by themselves. They're in there, and they just, over time, calcification of the tissue will occur, and they're just encapsulated here. And you can see the coiled larva among all the striated muscle here. So whenever you see a picture of a biopsy or this muscle tissue and a worm coiled up inside of it, you know right away you're looking at a picture of Trichinella spiralis larva. The infective stage of Trichinella is the infected larvae that are ingested. So how do humans get this? Well, we are actually accidental hosts. This is typically a zoonotic infection where it just continues among animals. But we get it when we eat undercooked meat that is infected with this larva. So a pig, for example, will have this larva in its muscle tissue. It's encysted in the muscle tissue of the pig, and if we don't heat up the pork enough to 145 degrees Fahrenheit, if we don't cook it long enough or get it hot enough, then the larva is still infective. It's still alive, and when we ingest that meat, ingest that pork, and our stomach acids break down the muscle tissue, we digest it, then the larva is freed. And then it gets into our circuit. So then the larva is in our intestines. It can leave our intestines, enter the circulation, and then go and set up shop in our muscle. So when this happens in nature, in the wild, this is how animals easily pass it from one to the next. It's when one animal dies and another animal comes along and eats it. So other animals that this can happen with are fox, bear, deer, moose. But typically of all those, we'd be more likely to eat the pigs, like I said, or deer, or even bear meat. So I always joke that once a human gets it, unless another human comes along, a cannibal comes along and eats our muscle tissue, or another animal comes along and eats us or takes a bite out of us, it's a dead-end life cycle once it's encysted in our muscle tissue. So in us, like I said, we ate undercooked meat, the larvae are freed, they rapidly mature into adults, and then the adults lay larvae. Remember, there's no eggs, and the larva go set up shop in muscle tissue. So it's a pretty straightforward life cycle. The diagnosis is very difficult. It's based on clinical symptoms, which can be very varied. It, the symptoms can mimic other diseases. It can be just vague flu-like achiness, the muscle pain maybe. There's usually a fever. Rising is eosinophilia would be a big clue. But otherwise, maybe just nausea, some diarrhea, swelling, blurred vision, general weakness as it insists in our muscles. And then, of course, history would be very important to note too. I mean, it would be very helpful to the doctor if you could tell them what you ate in the last month. But a lot of times, you might not know that you ate undercooked pork 3 weeks ago. That might not be something, that would stand out to you. Also are helpful, that's not listed on here, some other labs would be increased muscle enzymes in the serum. So examples of these are like increased lactate dehydrogenase and increased creatinine phosphokinase, and anything associated with an intense inflammatory response. So like an increased CRP or SED rate, something like that. Now, the most definitive thing would be to actually do a biopsy of the suspected muscle that's infected with the larva. Although that's a pretty invasive procedure and the doctor would have to know which muscle tissue to go after. So that's not the first thing they're going to do, is just go do a random biopsy. So this can take a while to diagnose. And there is a serology test, but it takes about 3 to 4 weeks to develop the antibodies to be detected for trichinella. So this concludes the chapter on nematodes.